All right, I think maybe we can get started since it's 9. So in the spirit of covering as many different kinds of human neuroscience data as we can, we're fortunate to have Kai Miller here to talk about electrophysiology data and particularly sort of advanced time series analysis methods applied to human electrophysiology data. And Kai, just kind of to introduce Kai, has done a range of interesting work on human electrophysiology data, also a neurosurgeon, so has kind of an understanding top to bottom of this field, this sort of human electrophysiology, and really done work all the way from models of the electrophysiological responses that we measure with electrodes at the level of cells and circuits, and all the way to these kinds of analyses that he's going to talk about today. And we're very happy to see to talk about this. Take it away.
Okay, thanks. Are folks able to hear me okay? All right, so I'm Kai Miller. I'm at Mayo Clinic. My background is originally in physics. I was a grad student and med student here at University of Washington. I did my physics PhD was in a building about 2 blocks away. This is the first time I've been here in 13 years. And then I also did a neuroscience PhD and I did a medical degree. So I have background that spans a fair number of things. Somehow it skipped to slide 8. And so what I think I can do that's useful for the group here is give you some idea for, we always have to have this for medical ahead of time because of financial entanglements, especially if I'm showing some things. But to try to give you an idea for what we do when we implant these electrodes in people, and why we do it for the setting where most folks record data. I can talk about others. I do surgery for children and adults, more children than adults, and I treat a lot of patients who have epilepsy, and those are the data that I'm going to show you today, are data that we recorded with epilepsy patients. We also do a fair amount with patients who have movement disorders like Parkinson's disease or essential tremor. And but today I'm going to focus on the epilepsy patients, and we also do some recordings with patients who have tumors that we take out. Throughout the talk, there are going to be QR codes that point to papers for my data that I've had throughout the course of my graduate school, and then residency and postdoc, and now in my faculty job. All the data that I've produced is up somewhere. I'm going to give you a link to a library that covers a lot of my initial papers that we sort of wrote before there were repositories to put data up. And so those are all covered under repository now, but now we try to put our data out with each paper in the way that I do things. And I ask that things be done in my lab with my graduate students and postdocs are rather than having, you'll hear from my partner Dora Hermes tomorrow, where she'll talk a lot about standardized data formats. What I typically do is pretty much the exact opposite of that. Philosophically, I think that, there's always room for different things. The big problem I have is that I don't have really the kind of time now that you need to have to relearn a new programming language well. All my code base from the last 25 years is written in MATLAB. So I've sort of stuck with MATLAB. I actually started out with Fortran, which probably most of you guys haven't heard of. In the 90s, MATLAB was mostly based on Fortran. The obvious downside of MATLAB is that it's proprietary and you have to pay for it. I don't think that's a great thing, but it sort of is what it is. And then when you go to access the data that we put out, you can either do it in BIDS format, and then and Dora really handles a lot of that. And then what I do is I put it out in MATLAB format with the code that I have. I feel like what I can do best is just try to describe the data to the best of my ability, and I limit the data down to, I try to have 1 or 2 data files associated with each patient that have salient variables. Please interrupt me at any point throughout this. For the folks online, same deal. Please interrupt me for any reason. And what I do is I try to have it so, there's a data that's kind of a time by channel variable, and then there's variable that would have, for example, EMG signals measured from the muscles, data that has the stimulus for what was shown on the screen. That's not going to be as relevant for most of the stuff that I talk about today, but if you decide to go to any of these manuscripts where you can get the data, or the libraries of data that we put out, that's the format you see it in, and they're associated Word documents that describe each of these things. Obviously, doing it that way is not great if you're trying to aggregate information across institutions and that kind of thing. And really, for that, or if you're trying to apply your methods most generically, for that, things like bids are great. You'll hear about that from Dora tomorrow. But my philosophy for data has much more been try to teach people how I was thinking about it, how to reproduce what I've done. And then the papers that we write alongside these things, I try to write out the math fairly well. And you should be able to reproduce. I firmly believe in reproducibility of everything that you've done in terms of sharing your code for how you do it. And I think it would be better if I used the most common language rather than MATLAB, which is what I use. So anyway, with that caveat, I'll get started. I'd like to kind of start out a little bit from clinical perspective. I think that's different from what you guys may encounter. A lot of the data that we record is from patients who have epilepsy. And so epilepsy is a disease where the circuitry of the brain, for one of a variety of reasons, is abnormal, and you have sort of a runaway positive feedback loop of electrical activity that results in a disruption of basically normal brain function, either limited to a specific area of the brain. If we hear people talk about focal epilepsy, usually that's what they mean, is there's one particular focal point in the brain where the seizure activity is limited to, or it can be generalized. And that means it's coming from everywhere. And a lot of times, what of those things it is depends upon whether we'll have a structural abnormality in the brain in a specific spot, or if they have something like, and that will give you focal epilepsy in many cases, or whether they have a genetic condition that results in all of the cells in their brain being sort of abnormal compared to most folks. When we're surgically treating epilepsy, before patients are ever considered for surgery, there are a number of factors that we think about. So the first thing that we have is imaging. Okay, so this is structural MRIs, principally. There are also things like PET scans, which look at metabolism. And we'll look for something abnormal. Here you see an abnormality in the part of the brain that controls the body, precentral gyrus. So we have imaging. And then we have electrical signals that we've measured from the surface of the scalp. So that's EEG, okay? There's also MEG, magnetoencephalography, but what the neurologists that I work with try to do is they wait until you have seizures, and the seizures electrically will look like something like what you see. See if I can get my arrow over there. Yeah, so something like you see here, and then that corresponds to sort of the left side of the head. If we're getting things to line up, which I tried to do. Anytime you're looking at clinical images, so if you guys are ever in a clinical conference of any kind, you'll look at it, and it's still most intuitive for me after doing this for however many years to look at it and say, well, the right side of the screen should be the right side of the head, but it's actually the opposite. And that has to do with the fact that when you look at somebody head on, the left side of their body is in the right side of your visual field, but it's still sort of annoying. So usually there'll be a big R somewhere on the image that tells you what the right side is. So I always do that to start out. And this is all I do all day, every day for decades of my life. So I think the convention they picked was wrong, but it is what it is. So here you can see some abnormality on the left side of the brain in the structural imaging. You see something on the EEG. And then the third piece of information that you hear people talk about is semiology. But all semiology for seizures mean is that's the symptoms that patients have before, during, and after when they have seizures. So in this case, I got Dolly to make me a picture of a kid with a shaking hand. Okay. The hand area of the brain is loosely speaking, almost probably close. Well, it's basically hand area is where we see the abnormality on imaging and where we see an abnormal EEG. And you say, well, these three things line up. So that's probably where this is coming from. And we can treat this in one way or another. And I'm going to show you something that is a relatively complicated diagram here. And I don't want that to be, this isn't something where you guys really need to look at it. You just need to understand that the clinical picture it is complicated, and it's because of that complexity that we have to bring patients in and put electrodes in their brain and monitor them for seizures. And that's the time when most people who do science with these data will do scientific experiments. So that's when Gabby, for example, will come into the OR and do tests with our patients. And so what happens is patients basically come in and they, you see on the top left there, they'll come in and they'll see a neurologist. A neurologist is a doctor who specializes in diagnosing diseases of the nervous system and treating them with medications. If they have a surgical problem, then they interact with me and we work closely together. So, you'll meet these individuals wherever you are. If you're a scientist that wants to become involved with this kind of research, there's a certain culture medically that you kind of have to learn a little bit about and mesh yourself in. But the important thing to remember is that, the clinical team, if everything is working appropriately, is sort of different than scientific collaborations where everyone's trying to establish themselves independently. What we're doing in medicine is everybody has a set of expertise and we have to kind of work together to do the best that we can for each of our patients. So patients will come in, usually they've seen some neurologist who is closer to where they live. Most of the places at the big universities are what are called tertiary or quaternary medical centers. So that means that, they will have seen not just a family doctor, not just an internal medicine doctor, not just a neurologist, but neurologists who, even in their community, who may specialize in epilepsy, and then they refer them to us and things get more exotic once they come to a big medical center like University of Washington, Mayo Clinic, Stanford, whatever institution you guys are most likely to be at, will have a tertiary quaternary center. And so we have a big committee, and it's called the Multidisciplinary Conference, and that's where you have neurosurgeons, neurologists, neuroradiologists, psychologists, psychiatrists. If you're a scientist who wants to work with these implanted human data, most of these things are online now, and I would just get on the list and start showing up and following this a little bit. So you start to understand the culture, you start to understand which patients you might be able to work with, start to identify what the epilepsy conference is. And they'll ask a question, and when I say they, it's me too, we'll ask the question, do we have concordance, which means agreement between the EEG, the imaging, and the symptoms the patients have, which is like what I just showed you. Okay, and when that's true, there are some special cases, like patients will have half of their brain will have died from stroke, that kind of thing, and I may go in and remove whatever residual tissue there is for a whole half of their brain. That's called the hemispherotomy. Not a good research setting, but it's something that we do. We'll also disconnect the two hemispheres. Those are sort of some of the more extreme things we do for epilepsy. But there'll be other cases where, like the example where I showed you, where if I take out a lesion that we see, or an abnormal lesion just means a structural abnormality in the brain that we see, it might make the patient paralyzed on half their body, that kind of thing. And if we don't want to do that, we put in a stimulator of one kind or another. I won't go too much into those. If you guys have questions, I'll be here today and tomorrow, and I can show you slides that explain all these devices that we put in. But today I really want to get to some signal processing techniques related to the diagnostic process. Okay, so if these things don't agree, or if we don't understand the extent of where the abnormality is, we'll implant electrodes. Okay, and that brings us down to this whole set of interventions that we do. And basically what it is we put implanted electrodes in the form of either stereo EEG, which is what you see here, or electrocorticography. And electrocorticography and stereo EEG are electrodes that we surgically implant in the brain. For electrocorticography, it means you implant them on the outer convexity of the brain. Okay, so just the surface that you see when you take off the skull and remove part of the dura. Dura is a leathery covering that surrounds the brain. And then stereo EEG is where we put depth electrodes into the brain. Stereo doesn't mean stereo like you have for stereo sound. It refers to stereotactic, which is the name that we have in neurosurgery for placing devices using a coordinate system inside the brain. Okay, and that's where the setting is for most of the recordings that I'm going to talk to you about today. So stereo EEG. This is the most common thing that's done now. When I started my research as a grad student here at UW, this really wasn't done in the US. And starting about 10 to 15 years ago, there was a transition throughout the whole country. And in fact, many places in the world now where people switched from electrocorticography to stereo EEG. This is what stereo EEG is. So on the top left of the screen, you'll see there's this bolt that's placed in the skull. It's a couple millimeters wide. And basically I make drilled channels in the bone and put this bolt into the bone. And then I pass wires through as accurately as I can into targets deep in the brain. And that may include cortex, white matter, basal ganglia, and thalamus. Okay. We want to understand not just where seizures may start from, but where they spread to. And that's sort of what these are. Gabby spends a lot of her time, and Lupita as well, co-registering the electrodes, finding out exactly how the positions of our electrodes relate to the underlying anatomy. Okay, and a lot of times we use fMRI ahead of time in order to try to get some understanding of the functional representation in that particular patient prior to putting these electrodes in. And that can be important, especially in cases where you have some pathology, some abnormality in the brain that has caused these patients seizures. And so, you have to take what you can get. The reason why I think it's useful to work with humans instead of, let's say, non-human primates is that I think that a human with an abnormality in the brain is a better approximation of the standard representation of human brain anatomy than is let's say, a non-human primate whose brain is 1/10 the size or something. So you'll see these images in other parts of my talk and probably in Dora's talk tomorrow. And that's just using the MRI from pre-implantation and registering it to the CT post-implantation to infer where the positions are and then plotting things. I think that it's important to always consider referencing of your data. So anytime you measure an electrical signal from somewhere on the surface of the scalp or inside the brain, you're measuring compared with somebody somewhere else. So the common references that people will use clinically are going to be the mastoid, which is just a bone that's back here. They'll put a reference behind the ear. Basically, that's good because there's not, if you're close to the ear, there's not musculature right there, or there's a little bit less musculature than most other places on the head, and it won't interfere with where your EEG electrodes are if you're doing EEG. We'll also sometimes put them on the chest. Obviously, the chest is a big muscle. You get a big heart response, so that's going to be cardiac contaminated. So heart trace is a problem. Or we'll do leg pads, a similar issue, and still, even if they're lying quietly, the heart signal is going to be an issue. Even though you may not see cardiac contamination directly in your traces, still the heartbeat and respiratory cycles, anytime you have a signal from the brain, whether it's hemodynamic with MRI or electrophysiologic with these implanted electrodes, it's going to be an issue. And so there are a variety of other places we can reference our data with respect to, and that's what you're seeing here. And if I'm doing deep brain stimulation, I have what are called cannulas. They're basically metal tubes we put in the brain, and we pass electrodes through those. I use that as a reference often. And for stereo EEG, a lot of the time we're referencing with respect to white matter, which we'll try to find some what we think is going to be relatively quiescent channel. But of course, you can pick up electrical signals from the white matter from passing action potentials, and that can contaminate everything else that you have, particularly for these stimulation evoked electrical potentials, which is what I'm going to get to in a moment. Re-referencing your data, there are a lot of different ways to do it. We've been working on a review of how you do it in general. I've been working with Alex Rockhill, who's a postdoc in Oregon, and he's actually participating in the course remotely this week, but he'll be in person next week. So you could ask him if you're interested in this type of referencing. And it's important to remember that when we measure these neural elements, whether we're looking at the effect of synaptic influence or we're looking at some secondary effect, like hemodynamics, the orientation of what we're measuring can be important. With fMRI, it's a little bit less important than, let's say, with stereo EEG, but re-referencing can actually influence your signal, as you see here. And that's really all I'm going to say about referencing today. The one thing I want to make sure of is if you start working with these types of data, understand that referencing, it's not like there's one best way to reference your data. It's that when you reference your data, that referencing process itself is a tool. So you need to understand what it is that you've done and understand what the relative drawbacks of whatever technique you've done are and what the relative advantages are. And oftentimes you're best off re-referencing your data in a couple of different ways and making sure that the biological interpretation of the measurements that you've made doesn't change as a function of how you've referenced your data in any kind of electrophysiology, whether that's EEG, MEG, or these implanted recordings. Okay, so that's all I'm going to say about referencing. If you guys have questions, you can always contact me or someone from our group. And now I'm going to get to the meat of what I wanted to talk to you about, which is this idea of probing brain networks to understand connectivity using these implanted recordings. So I'm going to have some illustrations that are both from the surface of the brain, which is electrocorticography, and from the penetrating electrodes in the brain, which is stereo EEG. And typically when you're working with these types of data, you're going to work with both because simply because you have the same surgeons that are implanting one or another, depending upon the particular patient. Although about now, in most centers in the US, about 90 to 95% of implants are going to be stereo EEG rather than electrocorticography. The idea behind this probing is that we will have two adjacent electrodes and we deliver pulses and we measure the deflections in the electrical potential at some other spot in the brain. And we allow several seconds between each one of these pulses. And the reason for that is that you've introduced a transient deflection and you want to give enough time for it to die out before you deliver the next electrical impulse. For people that are getting into electrophysiology, but they're used to fMRI, there's an important distinction here because in fMRI, you have this sort of linear summation of subsequent visual images that you've given. If you're doing evoked data, you can regress these things out. The same is not true with electrophysiology. There is a compound effect, and these things don't linearly superimpose with one another the same way they do with hemodynamics. When I started doing this kind of research, it was about 7-8 years ago. I ran into this problem that there was these responses in the brain. People were saying that there is a canonical form for the voltage in time, which is what you're seeing here. Does everyone understand what voltage is, what the time course of voltages is? So I'm going to be showing you traces where basically on the y-axis you have voltage. So that's how the sort of a charge deposition in the area you're measuring from changes over time. And time is on the x-axis, and we're looking at deflections over time. Typical time scales you're looking at are always going to be important. What they did in this paper is not usually what you want to do anytime you're dealing with electrophysiologic data or any data at all. Something your PhD advisor or your postdoc advisor should be hammering into you is the idea of always putting units on your plots. And so this was the form that all of the data is supposed to come in, right? And I was looking at this and this, I was going to see if this is true or not. And can I get a show of hands? Is there anybody here that works with ERP style data, evoked potential data? Okay. So for those of you that are used to doing that, you're used to a standard shape in the let's say visual evoked potential. You'll hear people say, N200, P300, these kinds of things. These are all things that people identified by looking by eye at their data. And I think that's where this came from. It came from an era when people were looking by eye at their data. But we're now in the age of computing where we can automate stuff that would have been impossible to do 30, 40 years ago. And the first thing I'm going to show you is a technique that I think somebody should have come up with 25 years ago when the computing technology came around that would allow us to basically just allow simple analysis techniques to describe our data for us. So for these stimulation evoked potentials, what we do is we deliver these biphasic pulses of electrical activity and give several seconds in between. And we might deliver a pulse here, and then we measure a set of responses that we get from that pulse out in another site. And we might measure a different set of responses at some other site. But if we look at just one of those, we can overlay some number of trials. We typically pick about 10 to 15 trials. And when we look at the average, people have called that cortical evoked potentials, or CCEPs. In our case, that's often a misnomer because we're not necessarily stimulating or not necessarily measuring from cortex. A lot of times white matter, thalamus, is basal ganglia. And if we look at some of these evoked potentials that we get from stereo EEG across just 5 stimulation sites, this is the family of responses that we get here. So the problem that I ran into when I started to do this is I looked at this set of responses and I looked at this canonical shape that we're supposed to see. And there's no way the data we actually measure can be explained for by this standardized shape here, right? So the field was basically going down something where somebody picked a form by eye and they're trying to force all the responses they have into a parameterization. And that's what you have. Anytime you say there's a shape that should be there, that's a parameterization that you're imposing on your data. And there's no way that you can do that. And so what I wanted to know is if there's no standard shape to look at our data in, is there a way that we can just understand if there's structure in our data without having to look at it by eye, okay? And still, I've noticed in the literature, the most common practice for any kind of evoked potential, whether it's EEG, or it's event-related MRI, or stereo EEG, or any kind of evoked potentials, is people will talk about a particular value that they get in their data at a particular latency compared with the event that they have. So in this case, the event is electrical stimulation, but if it's visual, that really relies on an assumption that you've made that you think that you know what that interval should be. And I would propose that it's much more important to capture the shape of the response and characterize that, okay? And there's some assumptions built into the way that I'm proposing to do it. Provided that you don't have significant time dilation, that kind of thing, I think it's appropriate. And so what I'm going to show you is a general strategy. And what I'd like to talk to you about today are 4 different things. And the first is a set of paradigms for how we can understand this electrical stimulation. Okay, because we have whole families of responses in these stereo EEG or electrocorticography data, and these measured data where we basically have a large number of electrodes we're stimulating at, a large number of electrodes we're measuring from, and different shapes associated with each interaction, okay, with each stimulation measurement interaction. So if you have 100 electrodes that you're measuring from, which is a lot less than we normally have, we usually have of order 200, 100 sites where you're stimulating at and measuring from, you're looking at 10,000 comparisons there. Okay, so that's not something you're going to do by eye, and you shouldn't be doing in the computing era. So we need some set of ways to, if we do want to think about our data in principled ways and isolate our perspective, at least temporarily, into one of a few different paradigms. And the paradigms we proposed were these. The first is what we call the hypothesis pre-selected paradigm. So this is where you basically say, hey, I think that there are different, 2 different sites, or maybe 3 different sites, but there's just a couple of sites. And here you can look by eye. There's a couple of sites that are important. I want to know what the interactions between these are, and I predefine them either based on anatomy known ahead of time, or MRI ahead of time, or in a lot of cases like a localizer experiment. So people that do fMRI will be used to this because you have this explosion of comparisons you can have. They'll do staged experiments where the first stage localizes down to just a few voxels you want to look at, so your statistics don't explode on you. And that's more or less what this would be. You have hypothesis pre-selected or another experiment pre-selected. Okay, so that's this paradigm. An example of this is some great recent work by Gabby, who's here. And what she did was she measured from the posterior cingulate cortex, and this is measuring from, I really don't know what the name of it is. It's either Pepez circuit or Pape circuit. I've heard people say both my whole life, and I don't know which is correct. But in this case, you can get a measured voltage here. And again, this is an illustrative slide, so Gabby knows to always have units, but she does have them along the y-axis or the x-axis. But you get some characteristic response here when you stimulate hippocampus in the posterior cingulate. But then you can also stimulate in the anterior nucleus of the thalamus, and you get a different one. And you can say, well, we've measured a significant response here. And actually, there's something really interesting where it looks like the shape is conserved, but just delayed by about 40 milliseconds when you're measuring from the posterior cingulate if you stimulate at the hippocampus rather than at the anterior nucleus of the thalamus. So this would be an example of a hypothesis pre-selected approach. There's also the all-to-all. Okay, so it would seem like this would, and for me, this is not this is not how I want to do science. I would recommend this is not how any of you guys do science. The way that people have done this in the past when they apply the all to all is they assume some form that every response should have. And then they say, well, we're just going to measure this. So we'll measure like the voltage at 200 milliseconds after we delivered our electrical impulse, and we're going to do that everywhere. And that's going to give us some global solution. And that's where you wind up coming up with where you hear things like, well, you know, the response at 200 milliseconds is perception and the response at 400 milliseconds is decision. Like, this is not disciplined neuroscience. You don't want to be doing that. So I would propose that for the most part, you don't find yourself in this regime, whether you're doing functional imaging or evoked potential studies like the ones that we're talking about. You can do something called the divergent paradigm. And what that is where you stimulate one brain site. You say, well, okay, if I stimulate here, and for those of you online, I hope you can see my cursor here in you know, something that is, you know, let's say face area, or if it's a little more interior, you have speech in the ventral premotor Broca's area. And I can measure one response, let's say out here in lateral prefrontal cortex. And then, you know, in response to the same pulse, I get a different shape out here in parietal cortex. The problem that you have is that the laminar architecture, the organization of the microcircuitry in each region of the brain is very different. Okay, and this probably influences your hemodynamic response as well. And I think that over time, people are starting to appreciate that more and more. But you run into the problem that different shapes that you measure in these different places don't necessarily mean that the way that two areas are connected are different, right? It's just that the architecture of the pyramidal neurons that produce our main response is very different in the parietal cortex compared with the frontal cortex. And so even though the response shape we measure in time is very different, we don't actually know if that interaction is different or not. So this is a little bit difficult because the same output shape in different areas may actually refer to the same thing, or may refer to different things. We have no way to constrain ourselves. And I'm going to come back to these paradigms later in the talk. A different paradigm is one where you say, hey, you know what I'm going to do instead is I'm going to focus on just measuring from one site, and I'm going to see what happens when I stimulate everywhere else. And here you have the advantage that if you get different shapes, because you're always measuring from the same spot, these different shapes mean that the type of interaction that this area you're measuring from has with those other areas you stimulated are fundamentally different. Okay. And so we call that the convergent paradigm, where different response shapes imply different input types. So now I'm going to tell you about a general tool. I think you can use this for any type of event locked brain data. Okay. And we call this canonical response parameterization. And the example for electrical stimulation that we would have of this, and although it would work for any set of repeated events, you could use it for HRS in response to visual stimuli. You could use it for peripheral stimulation and measuring EEG. You could use it in a lot of settings. But anything where you have an event and then you have something that happens after. So here we have a recording and stimulation pair of sites, and we measure an evoked response here. The black trace is the average of 15 trials. And what we want to know is we want the machine, like our algorithms, to be able to identify whether or not there's significant structure in that response. And you might say, okay, well, I look at that and I know that's there, but keep in mind now we're looking at 10,000 different things. Okay. And so what we want to do is we want the machine to understand when it's statistically significant. So a trick we can do is we can take our 15 trials that we have here, and we can go, for example, for the first trace, and we can normalize it. Okay, so that means we sort of take its magnitude away. And then we take its dot product of that normalized trial with all of the other trials. And that's what you're seeing over here. Okay, and you get a cloud of data. And if that data is greater than 0 and statistically significantly different from 0, you know that you have a significant response. But of course, you don't do this just for your first trial, because there's nothing special necessarily about the first trial compared to the second trial. You could use your 10th trial instead. But in fact, we use all of the different trials and project them all into one another. Okay, and we have 15 different trials here. And I've shown you ordering, where we have these projections or these dot products along, you know, with trial along the y-axis here. Okay, but we can actually collapse across it. So we have just one column of data there. Okay, and because my axes start at something like 70, you know that this is a very strong and significant response. So this statistical measure is just telling us what we could see by eye, but it's discovering it automatically. Now, there's a problem with what I did here. Does anyone want to guess what the problem with, and I'll tell you where the problem starts. The problem starts right here on the bottom left. Does anyone want to tell me what the problem is? Okay, my axes have units on them. The y-axis is bounded by what we've actually measured. Anyone want to tell me what the problem with the x-axis is there for evoked potentials? I see a hand. You're just stretching. Oh, come on. Gabby or Lupita? Okay. The problem is that if I go to another site and I measure here, and where it says replace lamp there is actually where we have our significant response. But basically, why should 1 second be the appropriate frame of data to look at? There's nothing, I've assumed a time scale there that's going to be important. And these types of things should be discoverable by our algorithms. And I thought, well, how do we discover that? What's a good way to do that? Well, first we have to ask, what are the features of our data that we can leverage for something useful? And the feature of data that we have here that we can either get naively or we can construct by baselining is that 0 means something. Okay, and 0 means there's not anything going on. Usually, right? If our data hover around 0, there's nothing going on. We assume nothing's going on related to our stimulation. Okay, and so we can take advantage of that. And that's why we say there's not much important going on after about 100 milliseconds there, because our data then hovers around 0. So what we can do is we can take that projection technique that I showed you before. And if we had 1 second of data, like you see there, that cloud of data, like I showed you in the previous slide of projections, let's see if I can get my cursor over there for those online, projections is here, okay? And this black dot is the average of those projections. Well, there's no reason why 1 second was important. I could have sent in half a second instead, or I could have sent in 100 milliseconds instead, or I could have sent in, let's say, I think that's 50 milliseconds instead. But actually, I don't even know what the appropriate fractions of data to send in are, so I can just scan all time points and I get something here. Okay, now what we have, if you think about it, is this dark black trace is telling us this is the profile for how my data is significantly different from 0 in terms of the reproducible shape of that evoked response. Okay, so that if I look at my evoked potential on the top right there now that you have on the bottom left, and I say, well, where was the peak of that response there? I see that the peak of that response tells me when my data starts to dive back into 0. So that tells me when it's significant. And that turns out to be a really useful tool. Because if you're parameterizing your data, you're trying to understand when it's significant and you want the whole shape, you don't want to include a bunch of junk in there. And that's what you would do if you carried it all the way out to a second, or let's say you thought 738 milliseconds was important or whatever. You shouldn't pick that. The machine should pick that. Now, I picked the peak of this distribution, but I could allow the peak plus some variance to take a little bit more time if you think that it should get closer to where the average trace crosses 0 of the voltage. You can have allowances for that kind of thing built in. And you do that by taking properties of this projection trace, this dark black trace here in the middle, by taking properties of that. So you could take, maybe when it's 98% of it, it would give you some width. And you'll see error bars on these in subsequent plots. So here on the bottom right, you see that because this trace gently approaches 0, your error bars on your projection are pretty high, right? But I thought for this initial parameterization, I'm going to just take the peak of that projection trace. And then I can parameterize the response, where I now take my individual responses, voltage is a function of time, and I can take an individual trial here, like you see, and I can say that voltage is explained for by this shape, C of t in red. That's essentially what portion of my data is explained by the fact that I gave an electrical stimulation, and scaling it by just a number, so a scalar. So this alpha is just a number multiplied by this shape, plus a residual term, epsilon here. Okay, and I can do that for individual trials. And what's really neat about that is that now we have something that essentially is telling us about our signal related to stimulation. The magnitude of the response to stimulation is given by this alpha, and the residual left over, whether that's residual that's related to other brain activity that's correlated with some other ongoing processes or amplifier noise or that kind of thing, that's all bound up into this epsilon term that you see there. Okay, so we can get a number of really useful quantities from that. Like we can get what the explained variance in a single trial is due to the stimulation pulse that you've given. You could do that in bold responses on single trials as well. You could do that in any event lock data. Okay. And if you want to talk about the magnitude of response in units that make sense, this alpha, when corrected by the square root of the amount of time over which alpha takes place, if you divide it through, or the amount of number of samples, when you divide through by alpha by that, we get this thing called alpha prime, which is basically sits in microvolts. So for those of you that are used to quantifying some kind of response and you hear people talk about RMS, this would be like a pruned RMS, where the RMS is just telling you, so the root mean square of the signal, but it's just the magnitude of the explained shape that we know exists on a single trial basis from the electrical stimulation we've delivered. Okay. We also get signal to noise in there, and we can also quantify noise. And now we have something where we can compare shapes that are wildly different from one another from different parts of the brain. We can compare them in the meaningful way. We can say the explained variance from electrical stimulation in this place and this place, even though the shapes are very different from one another, is 95% in the top trace. That's this far right term here, and 66% in the middle trace. And we also can immediately identify sets of responses from our algorithms that are not significant at all. Okay, and we've done this in the global sense, not in the sense of we picked some time after stimulus and then found that it wasn't significant. This is just telling us globally it's not significant. And there's no assumed shape because all of this was extracted from just comparing individual pairs to one another, and not even comparing them to the average. So as your size of your number of samples shrinks, you're not introducing self-consistency, which is what would happen if you were comparing to the average. Okay. We can automatically reject artifactual trials. So this is a case where, let's say I measured EEG and I stimulated down in the brain, and I get these sets of traces that you see in panel B there. But when I look at my projections, I have all of the projections that involved this anomalous trial here are shown, when other trials were projected into it, that gives you the raw data, the red traces, the red dots. And when this anomalous trial was projected in the other trials, it gives you the green dots. So you immediately identify artifactual trials. You also identify the single trials that are most representative of the population statistic. And that would be this cloud of data here with whatever trial this corresponded to. Okay. And so after I've thrown away my artifactual trial, I then have a cleaner set of traces, and I can then calculate my response. And I use this in a variety of other settings. We do things like deep brain stimulation for patients who have Parkinson's disease or central tremor or epilepsy. In the case of patients with tremor, sometimes I put an electrode strip through a little burr hole that I make where I put DBS electrodes in. I stimulate the thalamus, and then I measure evoked responses here on cortex. Okay, and then we can apply this process there. This is obviously something where we would be living in a hypothesis pre-selected regime. Okay, so the take-home points from this, and again, the QR codes point you to the code and data for this, is that we use these semi-normalized cross products, which is what you've done when you normalize one half of a comparison but not the other, to identify when we have significant interactions. And then we generate temporal profiles of this interaction in order to identify the interval over which we have a significant response. And then we perform a parameterization. And then in the language of that parameterization, we can talk about things like signal to noise trial of individual cases. I also apply this to action potentials. So for those of you that are familiar with action potentials and spikes and that kind of thing, we do a lot of experiments with that from our recordings in the thalamus. I'm not going to go into that too much, other than the fact that we can isolate these shapes, we can remove them from the background, and then look at interactions between these spike shapes and the background field potentials, because we've taken them out in a principled way. I'm going to go pretty quick through this clustering. The math here for this is a little bit complex. For those of you that want to do clustering, I think it's a useful thing to see, but it involves non-negative matrix factorization to do dimensionality reduction. And I'm around if you want to talk about that. But I think just the idea of parameterizing event lock data itself is the main take home I want to get through to you. Yep.
Can I ask some questions about the first?
The canonical response parameterization?
Yeah, so there are some questions in Slack that I will
Yes.
read out to you. So this is Jack is asking, in your linear equation for voltage, what assumptions are made, if any, about the stationarity of the signals over time? Or is the time interval so short that it's safe to consider them as short-term stationary? Does that make trial comparisons challenging?
Well, so for the stimulation of evoked potentials, and actually, you know what? I think I'm just going to skip the clustering and we'll go to, there's a challenge that I kind of have, maybe somebody will want to do, where I will get to the stationarity. And that may make it, that I think that's going to address his question. So of course, I'm assuming that there are three principal assumptions. One, I'm assuming that the relative latency of the response compared with the stimulus that I've given is stable. I'm also assuming that that my data aren't temporally dilated. And then more generally, it would be that if you have a polyphasic response, let's say like the one that you see on the top right there, that the individual phases aren't dilated. Now, this parameterization, there's a field of statistics that deals specifically with parameterizations of this type. And it was from that. I have a collaborator, Klaus Robert Mueller, and it was through talking to him that we started parameterizing our data in this way. And that deals with temporal distortion and so forth. It's called functional data analysis. And so there's a whole subfield dedicated to parameterizations of this type and how you deal with non-stationarities. I assume that I'll show you a treatment where you can get rid of at least the latency to onset assumption, which is particularly important if you're doing evoked potentials in the brain, because you deliver some evoked potentials in response to an external stimulus. So not to electrical stimulation, but let's say to visual or auditory stimuli, where your response you're measuring kicks in, more than 100 milliseconds after stimulation.
Okay, a couple more questions here. In that equation, to what extent might there be or not any effects of autocorrelation? Adding some additional auto-regressive terms in there.
So what I would say is that autocorrelation, the purpose of this is to identify statistically significant structure, not to tease out any of the nuances of that structure. So anything that metrics for autocorrelation, like for example, Fourier decomposition ahead of time would give you, I think that's second order, and you would hope that the statistics are wrapped into just a simple dot product. There are much more sophisticated things that you could do with this, but I think actually the power of this technique lies in its simplicity. And my big soapbox I want to stand on is, in no way should people be, it should be the exception rather than the rule, that people are looking at some shape by eye saying this is a P300. Let's look at the voltage 300 milliseconds after the onset. And instead, we should be letting our algorithms identify the structure, identify artifactual trials, and giving us something that is, in some sense, both parameterized in time and also parameterized in magnitude, which is what this gives us.
Okay, last one.
Yeah.
I think you just answered that, but are the data filtered in frequency in any way?
I don't usually, if your data have some DC offset, it's a problem because zero has to be important. So I think it's useful to apply a high pass filter to your data. And that could be, you just want to make sure the time scale of your high pass filter is much slower than the width of any of the variations in the polyphasic responses you might want to capture. Because if they're not, then you just gotten rid of everything. it gets a little bit tricky because a lot of times there's stimulation artifact, which I didn't get into here. And the way that I get around that is I do all the parameterization starting about 10 to 15 milliseconds after I deliver the electrical stimulation, because that gets rid of the stimulation. So, yeah. So for the folks online who are questioning, please correct me if I'm wrong. What kind of signal to noise are we dealing with? What I can tell you is that it's very highly variable. And, you know, we have signal to noise ratios here of let's say, well, we have 4.5 and 1.6, but we do see things that are very clearly real, and we can extract signal to noise. We can extract structure when we can extract structure with only 10 to 15 trials when our signal to noise is as low as like 0.5 to 0.6 with this technique. Now, if your signal to noise drops a lot, you can, I mean, you can do what you always need to do, which is you need more samples, right? And so you can get lower and lower and lower and still apply the parameterization technique if you have a lot of samples. And getting the ship wheel symbol, which is not good. So for folks online, if this doesn't resolve, what I'm going to do is I'm going to unplug the display and then plug it back in, but I won't disconnect from the internet. If I go to another window here. Okay, I'm going to shut down the application and restart it. We're on slide 28 there. Okay, so I'm going to skip past the clustering technique. I'll have QR codes for the papers that show you how to, for the paper that shows you how to do it, along with sample data. In lieu of something that I think would be fun if anybody here wants to try it. So we have this process whereby we identify when a signal is different from zero in order to get its latency in a naive way, latency from particular onset, and then we parameterize our response. Okay, that allows us to capture different shapes like what you see. We then parameterize our single trials, and then this set of quantities allows us to talk about our responses in ways that are somewhat independent of what the actual shape are, right? So if you say, hey, I want to compare, I know I keep harping on the N200 and P300, but if you want to compare them to one another in a quantitative way, this lets you do it. And so the challenge that I have for, and actually, well, I'll come back to it later. The challenge that I have for the group is this. This is something from my PhD in neuroscience, where we implanted what are called electrocorticography electrodes, so the brain surface pads on the fusiform gyrus amongst and other infotemporal areas, and showed people simple pictures of faces and houses at the same rate that you see here. Okay, and patients were supposed to tell us when they see an upside down house. And what we could do is we could then look at particular electrodes here. We measure the voltage. You know, we could see what happens with faces and houses. That face is my favorite face. She'll be talking tomorrow. And you have these event-related potentials where you get a very different voltage deflection with faces in blue compared with houses in pink. Okay, and we can look at the power spectral properties of this as well. And I'm going to skip past that in the interest of getting to the challenge that I have for you guys. And I'm sorry because this part is still rough, and that's why I think it's a fun challenge. So here we have a face response in red and a house response in blue. And what we do is we can then apply this parameterization process. We identify artifactual trials and remove them. And then we have our remaining trials left over. And what we can actually do is we can jitter. And for those online, I don't know if it's showing up on OWL, but you can jitter your individual responses where you get the overlap much in the same way, but a pair to pair, not trial to average response, much in the same way that we did for individual trials before, but now we're jittering them in time. So we're allowing for differences in latency by looking at the overall overlap in the global shape, meaning global over time, not global over trials, but global over time between two trials. And then realigning each trial compared with the average latency between it and all of the other trials. And so that essentially sharpens our response that we get there. And then we can first focus on the top. We can apply this parameterization after we've jittered the trials from the time of visual onset, and that's what you see on the top. And then after we've done that, we run the process in reverse. So we run from the time that their significant response ends, and we run it back towards time zero. And what this does is it identifies the onset of the response. Okay. And so we've now run this parameterization process forward, where we do these projections as a function of time forward. And then we reverse the process and run it backwards. So we have the onset and offset time. And so what you notice here is that basically in this bottom trace, the yellow is the time of parameterization. And you can see that rather than doing what we did for electrical stimulation, we're now doing it for an evoked potential, much like was asked online. And you get the onset and offset times. And I apologize that these are just written out, but you get something interesting. For example, the explained variance now, instead of being 84%, is 90%. And the signal to noise ratio jumps from about 2.5 to about 3.4 by ignoring all of the junk data before that area of the brain reacts. Okay, and that's in this parameterization here. I should have put that up. And so the challenge that I would have would be to take a data set, and this data set is part of electrocorticography library. It's online. I'll bring that back up. I'll bring that icon back up for you guys, and to apply it to these simple face and house data from implanted electrodes, where you have a number of different electrodes that you're measuring responses to, and really apply this parameterization process to visual evoked potentials, because so far we've only been applying it to the stimulation of evoked potentials from inside the brain, the electrical stimulation of evoked potentials, and to action potentials. There is a key variant here, and this is a technical nuance that I think makes the problem interesting for people that are learning. So anyone in the audience, raise your hand and tell me what's wrong with the trace in the bottom left for the significant interval that we didn't see in the previous slide. Oh, come on. Somebody must see what's wrong. What's wrong with this here? What's that? Yeah, it misses this first deflection. And if we want to understand why it misses that first deflection, what we see here is that if we look at our profile that you have here, your projection profile, the peak of the projection profile actually comes later. And there's this initial phase in the polyphasic response that's missed. And what you need to do instead is take the first local maximum rather than the global maximum of the projection profile when running it in reverse. Okay. And it allows you to miss it allows you to get around if you've missed an early transient. And so I think this is a fun problem to work on. The code are out there. I've written the code to do the forward and backward, but I think it's a fun challenge for people to reconstruct that from the data that are out there. All of this stuff is in Python through the efforts of a number of people, including Alex Rockhill, who's here as part of the course. So you could talk to him about that. But I think that would be a fun challenge and would be useful scientifically for the field. So for anyone that would potentially be interested online or here, these are the two papers that describe the library of data. And for anyone who teaches any courses, as a grad student teaching undergrads, or for anyone who wants a library of implanted human data you could use for courses, or you could use some of the data sets from that have never been published. The scientific contents have never been published. And then the bottom right is the original paper. But that was even in 2015, we didn't have great repositories yet. So so those two papers have the data, and then the code to do the parameterization is here. And for anyone who's interested in sort of getting into these epilepsy data and you want to understand it clinically, that's in the QR code at the top. And I'll be available to work with folks for the next 2 days if they want to do this as a small hack project. And I'll be available virtually next week, but I'm operating most of the week, so it'd be at odd hours for you guys. Yeah. So, yeah, question?
Yeah.
So that's what that challenge is. What she asks is, have I gone back to old data sets and applied this technique to old data sets? So this would be applying it to an old, basically face house localizer experiment from implanted recordings. And those were done, it sound really old, but between about 10 to 20 years ago when I was here as a grad student at UW. So yeah, that would be this project here. Would be applying it to these face and house data. So it's this stuff would be it. And I can also provide the slides to anybody that wants them. Yeah.
Yeah, thanks for the great talk. It's really interesting. Can you elaborate on how this compares to cluster-based permutation analysis?
How it compares to cluster-based permutation analysis?
Yes.
Well, I mean, what I would say is when you say, well, I don't know what that particular thing means. It sounds like from the name that you generate clusters of data that you think are representative of some underlying distribution, and then permute through those to see if your answer changes. Is that right? See if your clustering changes.
Right. It's somewhat different. The future people do that, or the way of that. So it basically just looks at like with the most significant cluster in any point of data across time or frequency.
Well, I mean, I would say that without knowing exactly what that algorithm is that's in the field trip library, what I would say is that you certainly get for single trials, you get in this process, you get clusters representing the relative importance of individual trials. And that's this is, I guess, just an example, but before I'm trying to get my before you've calculated the optimal time to do it at, but you have individual trials, and you see how well, I guess if you mean there's different behavioral states of some kind or different responses of some kind, and you're associating those with clusters, you could apply a second order clustering on top of the parameters that you get for individual trials. You could apply that to this, and then you would be applying a clustering algorithm to that. There is clustering that is done that might be more similar to what you're talking about, and that was this other topic I was going to talk about, the basis profile curve technique, and that identifies clusters with some non-negativity constraints using a somewhat similar approach to this with non-negative matrix factorization, basically. So I don't know that particular technique. I don't know what that buys you. What this buys you is it buys you a succinct representation for a potential interaction that's captured by a set number of time course trials with their event log. And then you can use that parameterization for whatever kind of clustering that you want. And it's the idea that each, let's say, interaction between trials, or the amount at which trials are similar to one another, is always done in a pairwise fashion. Those would be the unique features of this. I don't know what that other technique is. But you could do, in some sense, the way when you reject specific trials from this, that is a type of permutation, because you then get rid of it and rerun it. But you could apply a whole variety of techniques on top of this. You could think about this as generating a feature space that would then be used in the clustering algorithm, if that's your goal. And that's what's done in that basis profile curve paper, except that takes advantage of the fact that you have some hierarchical structure in the data that you're looking at.
All right.
Yep. Thanks a lot.
Thanks, Kai.